Focus, abracadabra.
Abracadabra, we are live Saturday night at the Atheist. And we have a nice little icon. Mutable's here. We have a nice little icon representing the creationists coming to get me, coming to take me away with their pitchforks and their torches, as they did last night. So that was probably as famous as I'll ever get with them. Hopefully. It was actually kind of disconcerting because every time I tuned in one of their streams, they were talking about me. It's like, geez, what did they do? Huh?
How many streams did they have?
Well, they had 2, but on all 3, the chat they were talking about me. So they had a stream. Let's see, they had a after James tour, and then Standing for Truth, of course, set up the debate that I never agreed to. So. Anyway, I'm going to be talking to Tangelo, I think, at 11 a.m. on Monday morning on this channel. So, and it'll just be hopefully a pleasant discussion about why we believe in evolution or don't. So, kind of nice. That. So,
okay, I just looked them up. They had a total of 14 and a half hours of streams last night. That's insane.
Oh wow.
Yep.
Because they had 9.5 hours on John Maddox's channel, and then about 5 hours on Standing for Truth.
Was it that long? Or did Maddox do one that I missed? Let's see, the after show.
Well, no, the James.
Yeah, 9 hours.
9 hours.
Yeah, and during that 9 hours, I think I was mentioned in chat 282 times. So
popular.
Yeah, worrisome. So. We had to put in some extra security guards, and we bought a couple of Dobermans.
Oh, cool.
Yeah.
Security raccoons.
They're out there. Yeah, we got security raccoons, rabid security raccoons, mind you. So
hey, that reminds me. Have you guys ever seen like this one particular movie where it's like, are those sharks with lasers on their heads?
Oh no, I haven't seen that one. I did watch all the
Powers or something.
Yeah, I think it's that. I haven't seen it, but I know that clip exists.
Yeah. What were you saying, this Speed?
I watched the Sharknado movies, and Zombeaver was pretty good.
Yeah, I still have yet to see Zombeaver, but I've seen Sharknado. I've seen Birdemic. The Room.
Birdemic, I have not seen that one.
Really? That one's a classic.
Oh my God. Killer Tomatoes, a classic.
Has anyone here ever seen any of the Godzilla movies by any chance?
Oh yeah.
I personally have not. I know Dapper Dino is quite the fanatic on those though.
Oh, cool.
I used to watch those when I was a little boy, and I was like, what the fuck? Colbert did a thing. There's some blue hole in the ocean or something that they're going to explore, and Colbert was saying, no, it's 2020. Godzilla, do not go. Step away from the blue hole.
Pacific
we don't
Rim.
want to go there. I thought of a solution for our leadership problem in Washington.
Oh, really?
If Hollywood offered Trump a sitcom where he could be the president for 4 more years on a sitcom, he might take it. And they'd have to convince him that it was a reality TV show. But I think that they could do that, and it would work.
Yeah,
we get rid of them. I don't know. Just a thought. So, Immutable, what the hell are you doing?
Not too much. Relaxing, chilling.
Chilling. You said you.
Oh, sorry.
I'm not.
So anything exceptionally stupid come out of Maddox or any of the other clowns?
They've been quiet today.
Have you?
Really? Yeah. They've been quiet.
After 15 hours of streams.
Yeah. I think they're resting. I don't know. They came out and they bashed me on every video I did. For a while I had Smokey on almost every one, and I don't know.
I just don't know like how many things you can possibly talk about without, getting bored.
I know. I think they got bored with me after about like 45 minutes in their stream. or 30 minutes maybe. But they decided I wasn't showing up. Well, the chat didn't get bored, but they did. They threw Ian Chen out of the chat.
Really?
No,
What's he even saying?
I don't think he said all that much, but I think he was having a thing with Slam RN or something.
Yeah, I've never seen
really?
Ian to be like disrespectful or anything.
I know.
I mean, he does kind of like make fun of people. He's not like super polite, but I mean, like he's never like openly rude. You know what I mean?
Yeah. Well, he was a little put off by getting tossed out.
Really? Yeah.
Yeah. It seemed to disturb him a little bit. Oh my God! I don't believe it. Oh, there they are. My bookmarks. Looking at my bookmarks for this video, I found an interesting little piece down here at I think 109 that might be worth taking a look at if I can share a screen. And I will share a tab. I'm getting better at this.
Did you share audio?
I always share audio now. I'm really good at that. I'm really like for the last 5 times I've done it.
He has become specialized.
I'm learning.
He has officially learned a new skill.
I've learned a new skill. Now,
do you watch these like hours at a time? Oh, what'd you say, Andrew?
I do.
How many points can you rack up?
Yeah, I would like somebody else to start watching these things and bookmarking them.
That's a lot. I don't know. I watch like 15 minutes and then I either get bored or pissed off and then I leave.
I started running them at 2 times speed. You know,
fair enough.
And it makes it a little easier. But then you got to break and bookmark and back up, and that becomes kind of a problem. So,
yeah.
But I found this. Let's see if you guys can hear it. Let me know if you can't.
He would have to permit it a certain way, or he'd have to judge it instantaneously in order to maintain his righteousness. So by creating a reality within the scope of his certain nature of morality, he has to kind of follow its own rules. He sets the rules and he follows his own rules. Otherwise, he basically makes himself a liar. You get what I'm saying?
So I'm going to hell for rules I transgressed on.
Well, don't put it so specifically like that, because that's not essential. When
I'm trying not to sort of like,
you will suffer exactly as you cause suffering in your life. Yes, absolutely. Yeah, and you will be burdened by the weight of knowing you could have avoided it. Yes, absolutely.
So,
and if you caused anyone else to end up in that pit with you, yeah, you'll probably, if in a just universe, I would imagine probably get a little taste of that yourself, probably.
Sorry, I thought a belief unless you're holding a gun to somebody's head.
What's that?
Sorry, I thought belief was kind of like a personal choice. I mean, unless, I'm holding a gun to somebody's head and like just telling them, you better believe what I tell you or else you're going to die, and then kill them once they, how would you coerce into accepting what I tell them to think.
Oh, no, if you preach evolution in school and you teach children and they buy it and they stop being good Christians, then they will go to hell and you'll have to go to hell for them.
I think that was an interesting claim. The whole thing, like you'll experience as much suffering as you've caused. Does that mean that I'm going to be stuck having internet conversations for all of eternity?
Yep.
Apparently.
Could be bad. Okay.
In hell, did we?
Oh, yeah. Do you imagine any type of justice without it?
So,
just
how would Hitler, Stalin, Mao, Pol Pot, how would any of these people ever have any?
Okay, he's back to Hitler again. You hear that?
Yeah. Hitler's like his favorite person in the world now, apparently.
Yeah, and like he can't imagine a universe where Hitler isn't like suffering right now, currently. And I don't know. I'm kind of pleased with the fact that Hitler no longer exists. I don't know about you, but
I'm fairly glad that he doesn't exist.
I was ready to let him go. I don't know. I mean, we don't need him down there because what if he got out?
The incarnation.
Just imagine if you're resurrected from the dead. That'd be fucking insane. I mean,
zombie Hitler? That sounds like a movie that Speed would watch.
Zombie Hitler. Oh my, that would be good. I would watch that.
Type of justice without a hell. How would most?
Oh no, Hitler, Pol Pot, and Stalin in like Centipede Dictator or something. Human Centipede Dictator. I'd
Zombie
watch,
human centipedes.
watch that.
Psychopaths ever have justice without a hell? How about the guy who murders his family and then offs himself right afterwards? What type of justice will he ever have?
Well,
You
what
know,
about
I mean,
what about the guy who?
Can I say something?
We,
Okay,
Go ahead.
psychopaths clinically can't have empathy, so they don't know that they're doing anything wrong,
yeah,
and then you're just gonna like torture them for all of eternity.
and they're gonna.
I
be
mean, I'm
like,
fine with like locking psychopaths in prison and stuff like that.
Yeah.
Maybe even like death sentence if they do something extremely heinous. But like torturing someone for not knowing that they were doing something wrong.
Well, I think
don't
when somebody commits a mass murder, it's kind of natural to, and then they kill themselves. I have this natural urge to want them to suffer longer.
Yeah.
Everybody does, and that's kind of what Smokey's talking about. So these Christians, it's part of their. Not only do they imagine that they're going to heaven, but they also imagine that all the bad guys are suffering. How juvenile is this sort of? I mean, really, come on.
And I think this may be my bias, but I kind of this is the whole problem I have with this. Okay, you're going to heaven or you're going to hell kind of thing, because I mean it excludes certain parties, and not only that, but like what about dogs or
Right.
apes or things like that? I mean, like I said, it may just be my own bias, but like I think like this exclusion, because like if what about natural disasters? Like, are we going to count those? Like should the tornado go to deep
should
down?
suffer.
Basically, should the tornado suffer?
And I'm kind of curious. I'm kind of curious in this aspect of like, oh, you will have done to you what you've done to others. I mean, like, okay, Smokey, so like if you fucked over people like financially or other shit like that, you're like never repented to them. Like, say they died the next day or something
Right.
like that, like where you weren't permitted the opportunity to like apologize to them for what you did.
Well, those
Like-
white.
Are you going to hell as well? I mean, what becomes of that? Like, since you could not correct, you know.
Well, I
like-
think because he's. Well, I think is it Pete Tango that's talking to me? He addresses this.
He's just going to the electric chair and he just goes, and he's done the most horrendous crimes.
yeah, and he's gonna probably go to hell.
Is he? Is it, or is he going to? And he says it sincerely. He's raped and killed children. He's killed families.
Sure.
And just before the electric chair, he goes, "I ask for forgiveness."
Well, I don't really believe.
If he truly believes, if he truly believes in his heart and
Hold on.
he's truly sorry, then yeah. But no, if he's just saying it to be a cheeky smart ass, then no, he's going to go straight to hell.
Yeah, and just to clarify too, you know, there are most people, and someone like that you're kind of describing, you know, it's a little bit easier for us to reconcile that as someone potentially a Romans 1 reprobate. For him to get to that point, you know, like
again?
right before he's actually
The reprobate again.
What did he say regarding that though?
Something about
It's an interesting concept. I think I sort of got the idea of it whenever I had a super short conversation in a comment section with them. It's a concept known as judicial hardening, I believe, which is basically like God hardening the hearts of people who aren't truly Christian but are only asking for forgiveness for like selfish reasons or something like that. I don't really get the concept. That's not really something I had ever been taught in Sunday school or anything, or
Right.
a concept I'd heard about.
I heard him talking about it the other day because Nicholas apparently had the wrong idea of what a reprobate was, and they
Really?
three of them were just went nuts on him, kind of like they were really concerned and they had to solve this problem, and that's when I got this weird feeling that these people were like aliens or like they belonged to some weird club that I didn't really understand, and. I mean, it's really odd. I don't have that clipped, but maybe I can find it again. I'm not sure if I want to. It kind of spooked me. It kind of freaked me out. When they start talking to each other, you know, like brother, and they have these like arguments among themselves about some biblical passage, it kind of freaks me out. I mean, it's like, it's like you walk into a universe or like an Illuminati meeting where you have no. I mean, it's just really weird, and I thought it was strange. I don't like it.
Executed, and did that actually be the moment that he repents? Number one, I don't know how feasible that is. Number two, I don't actually have any scriptural backing to believe that that's the type of repentance that would be accepted.
But
Because
it
the idea of
Yeah, I'm not sure where this goes after this.
We're about to find out because Smokey's about to lecture us on it.
Oh, you want to find out?
Sure.
repentance, the idea of repentance in the terms of the Christian faith is to turn from your sin to actually do different, to make up your mind different. Like your positioning to never change up until the moment of your death. And like, say you see a bus coming at you and you've lived a horrible life and you're like, well, if there's a God, please forgive me. That's not how that works.
Well, yes,
Really? I mean, that should work that way, right? I mean, if you really mean it at the bottom of your heart, I don't know.
I guess there's a lot of people that are going to hell then, because I mean, a lot of Christians have done like sudden deathbed, have done like deathbed conversions for other people, and wow, okay, I guess they're all going to hell.
Yeah, it's like if I convert 5 minutes before and I don't have time to commit any sins, and I agree, you know, my heart has changed, I mean, I get to go to heaven.
It's like a point system. Basically, you get too many points against you, and the ones on the other side don't count. Basically, that doesn't seem very fair.
You know, that sounds very Catholic for Smokey to be saying. That's pretty much what the Catholics do.
He's very like, it's hard to pin down where Smokey is.
Well, he's definitely not Catholic.
Yeah,
Well, he doesn't like
he's not. It's just. Yeah, it's just hard to determine like where on the Christian spectrum he lies.
Yeah.
Like in which of the thousands of camps does he actually like align and or associate with?
I just now made a decision. I'm going to, like, if I know when I'm going to die and I know I've got like 5 or 10 minutes left, or even a day, I'm going to like get my mind right, and I'm going to make this heartfelt commitment to Jesus and turn my life over to God. Do you know why? Because all of those fuckers are going to be up there, and then I can be up there and I can argue with them about evolution for eternity. I like that.
Oh dear.
He has to actually believe. And so if he actually believed and he's getting all the way to this point, this is not how that would go down.
Well,
what's
the smallest sin, and I'm doing that in quotes, that will get you into hell?
Sorry?
Anything.
What?
If you don't believe, just anything. If you don't believe,
evolution.
Yeah, all flesh.
It doesn't matter how good, how perfect a life you live.
Okay, so let's just say that this dude sincerely, like, he's going to the electric chair and he sincerely says, I repent. With all my soul, I repent.
Oh, well, if he fully means it, then he'll go to heaven.
Stop, please. But at the same time, a soccer mum is going to work and she gets collected by a truck. And her last thought was, you know, I'm finding it really hard to believe in my religion and Christianity.
That's not practical, what you're describing.
Why is it not
It's grotesque, and it's not biblical, sir. It's
grotesque. It's not biblical. You're describing things that are happening on two potentially different sides of the world. That's not biblical, sir.
No.
But that's the most common kind of atheist. How many atheists do you know are just fucking serial killers?
Yeah, right.
This is the most common dichotomy. I know more soccer mom atheists than fucking serial killer atheists.
Yeah. Or people that just, you know, kind of lose touch with the whole religion thing and they're not thinking about it. I don't know.
Yeah. I don't know. This isn't a false dichotomy at all. Okay, whatever.
Yeah.
Okay, well, I'm telling you, because that's not how salvation works, and certainly that's not even how people's beliefs work.
What?
Yeah, Tango, this is really just silly because this is about something that's deep and long-term and spiritual, and you're making it about like quick moments of like a fleeting thought.
And you're making it where...
Okay, so these quick moment turning your life over to Jesus don't work, they're saying, in their religion? I don't know.
Apparently, I think they're trying to get him like to acknowledge that this is like it's a short answer thing when it can't be, or they're trying to get them to say something. He's trying to get them to say something unbiblical.
Yeah. Well, okay. Don't they always cite that there's one atheist who converted to Christianity on his deathbed, and he was like an atheist philosopher guy, and they always bring him up. So does that mean he's not in heaven either because he converted on his deathbed?
Was he the one that was kind of senile? He's kind of losing it.
I think that's a different one. The one that was senile, he converted back, or he like converted to Christianity in his like 50s, and he was using, he survived until his 80s. And he was like writing stuff like apologetics literature. I'm trying to remember who it was, but I remember someone converted on their deathbed, and everyone was pointing to it as like, "Oh, great salvation moment."
Right.
If it works for this guy who's literally spent his whole life trying to dismantle Christianity, then why doesn't it work for this guy who's been, at best, neutral towards Christianity and just been a shitty person?
Yeah. Where's the justice? And it sounds like these guys are kind of making excuses for all these fine point questions, Yeah. Like, I think they're trying to get the conversation done with because it doesn't make them comfortable.
Yeah.
I don't know.
Not the topic of our religion or of anything. It's just really.
Yeah. I don't know. I'm done with this. Are you done with this?
Yeah, sure.
I guess.
Yeah, I think one of the reasons they're trying to like avoid this question is it just sort of confuses their whole like punishment mentality, because really the only thing that's biblically like canonically punished is apostasy. Every other thing is forgiven through Christ, right?
What is apostasy?
Like not believing in.
Oh, okay.
Yeah.
That's the thing you get executed for in countries where these people are allowed to be in power.
Yep. Actually, Sudan, I believe, just got rid of the death penalty for apostasy like a couple of days ago. So
how modern of them? How 20?
welcome to the 18th century, guys.
Yeah. Yeah. Okay, I found this little gem here. I think this is the one. Boy, my bookmarks, my bookmark friend from Rocket Notes. Rocket Notes is a thing where you can tag these bookmarks, on things. And it's only like $2 a month, and you can have a database and everything. And I like the guy a lot, and he's really responsive, but he does have a little bit of a formatting problem in his latest release. But anyway, this is about the children. Let's go. Where are you? Oh, wait a minute. That ain't going to work, is it? I got to switch. My bad.
Speed, you are failing.
I can't handle failure. Okay, this one is Dr. James discussion open mic. I will click it. Got the audio. Huh? This is the one on Maddox's show that ran for 9 hours. Oof. So, are you looking at the right one now? Let's make sure we can hear this.
It's in super low quality for me. Can y'all read anything?
Nope.
Well, there's really nothing to read.
Okay.
We got Nicholas, Proclaimer of the Messiah. We have Smoky Saint. We have Jungle Jargon. And we have Tony. We know Tony. Is that Anthony? Maurice? Tony?
I can only assume so.
Okay.
Guys, seriously. If they're allowed to shoehorn naturalistic conjecture into the schools and somehow claim it isn't indoctrination, there's no reason there shouldn't be an intelligent design instruction also being taught.
You don't understand.
Right.
Okay.
Yeah. Here we
that
go.
makes me think that you don't understand what indoctrination is.
Right.
In a state of indoctrination is a state where you can't question anything. It's a state where you are forced to believe something, and any attempt to believe otherwise results in like a disciplinarian type of approach to correct that.
Mm-hmm.
And how many heartbeats would it take them to sign on for a bill where they were required to not believe in evolution?
Yeah, I mean, the thing is, like, the fact that science has conjecture and the fact we can be uncertain and that we publicly communicate this is one of the merits of science.
Yeah.
Because we can always correct ourselves upon new data being introduced. Whereas intelligent design, there is no way to falsify that kind of theory ever. And there's no way it can be validated or invalidated. So it has no value in that sense.
You know, personally, I kind of support the teaching of like Christian creationism in school, because the establishment clause of the First Amendment would also require us to teach like Hindu creation myths and Islamic creation myths.
Yep.
So, I mean, that in my opinion would be better justice than anything else.
I think it would be kind of cool.
Well, yeah, watch the Christians complain that now there's other forms of creationism being taught. That's not just Christian creationism.
Right.
I know. Yeah.
Yeah, let's see where he goes with this.
That is total nonsense to begin with.
It is. They should not be allowed to indoctrinate children with naturalism if they're claiming they don't like indoctrination.
My guy, you ever heard
Anybody
of
who is an abio?
So how the hell are they doing that exactly? By teaching
I don't know. They're presenting information, allowing students to ask questions about it,
read
and
science,
come to their own conclusions about it, and telling them that if they'd like to learn more, to read, articles in technical literature on it. That's indoctrination.
Yeah,
that sounds like a fairly common like college curriculum to me. Yeah. But that doesn't sound like indoctrination.
I mean, my problem with it is if you want to call testability and rationalism and things like that naturalism, however you want to call it, in some form or another, people cannot live without making quote unquote naturalistic assumptions by the logic they're using. Mm-hmm. I mean, if you don't go out and actually observe things and focus your assumptions on that, I mean, without even thinking about it, we make those kinds of assumptions. So that doesn't seem very logical to me.
Yeah.
Right.
Yeah. To me, teaching ID alongside like evolution and stuff is the equivalent of teaching like angels holding our feet to the ground alongside gravity, as like equally valid hypotheses. Like, come on.
Yep.
Or if this meadow, like this particular flower, started to go extinct, we would also have to introduce a possibility that God was pissed off at this flower and that he was intelligently intervening and causing its extinction.
One day he decides, I don't like the color yellow anymore.
Right. And we would also have to conjecture, allow the other group to hypothesize that it might be the witch down the street who lives in the ugly old house where the shutters are falling down, that she may have done it. Mm. So,
and they accuse us of basically like when you talk about ghosts, goblins. I like the example of gnomes with chewing gum connecting planets together under gravity.
Yeah.
And if like they accuse us of saying, well, you're going far-fetched with that. We don't believe that. No, you kind of do. It's basically equivalent.
Yeah. We just don't call it chewing gum. We not publicly anyway.
Right.
A Genesis supporter, they need to watch that video.
Totally.
Right. Yep.
I mean,
that's it.
Silence them. Yeah,
that's it.
What video? I'm not
sure.
sure.
All in a nutshell, right there,
man.
Right
there. It's straight to the point.
Moment.
Oh, the James Tour video. Oh. If you watch the James Tour video, you'll know. I mean, they believe this video was just the slam dunk, the last blow for evolution. They're pretty excited about James Tour, I think. Bless their little hearts. I'm not sure what else we had going here at 9:52. Not sure.
Whoever wants to break that down, Aaron Ra, speed of pudding, anybody.
That was just a speed of pudding mention.
Did the James Tour video mention anything about common descent or evolution, or was it exclusively focused on abiogenesis?
Just abiogenesis.
Okay.
I don't think Tour does that publicly. He doesn't come down on the side of ID.
Yeah, good. I personally doubt. I wanted to ask this, but I missed the live stream. but I doubt they would have taken questions about evolution anyways. But I was curious what James Tour's official position is on common descent, because I know he signed that Descent from Darwin letter, but so did a bunch of other scientists who agree with evolution. So
yeah, there's a lot of controversy. I'm not sure what this one was.
Absolutely. We'll get into some of the chemistry. Yeah.
Here's the tag team you're talking about, Smokey.
Well, I have a tag team coming up with Skylar Fiction and T-Jump.
Oh, that was on the debate. I don't think there's much else on this one that I tagged. So.
Is making excuses for not showing up tonight, saying
I
that.
see what.
Yeah, that's just about my not showing up for the debate. My excuse was I said fucking no. Fuck you. Yep. I said no. Yep. I never agreed to show up. SpongeBob Idiot Box. Let's see. What do we got here? Okay. I think it's back to this one. So where was I? 109, we did that. Okay, 2. So 155, they start talking about evolution a little bit, I think. Oh, wait a minute. I can't do that, can I? God, I hate StreamYard.
I like StreamYard. Why?
Well, I'd like them if they give you multiple sources so you could queue stuff up in advance and switch over real easy. Wouldn't that be nice?
What?
What is that?
I have no idea. Sounds like music. Is there music in anyone's background?
It's ghosts.
Yeah. It's got to be ghosts.
I hear things moaning and like making noises. I don't like it.
So if we lined up all the specimens of the skulls we have from the most ancient ones to Homo sapien, where would you point and say, no, monkey, ape, maybe human?
Is this the P-Tango guy?
Okay, well,
yeah.
Well, P-Tango, at this point, since we're going to be going around the room, you're going to have to make that a rhetorical question because we're now. Are you yielding your time? We're going to move on to the next person.
Yeah, go on.
Okay, so now we'll move it to C. Brown. Go ahead, brother.
No, maybe not.
Okay, so I mean, it doesn't make any difference. You know what? A creator created millions of different species. You're going to find some similarities in species, period. You're going to find dogs and cats got similarities. You're going to find human chips got similarities. You can't come up with that many different type species and not have underlining type of characteristics.
Wait,
just like
can I
they
make a point
want to
about
come
this
with the story about.
Yep.
Okay. Yeah, humans and chimps have a lot of similarities, and humans have more similarities with chimpanzees than gorillas, and humans and gorillas have more similarities with each other than orangutans. And that's the idea of phylogeny. If we had shared derived synapomorphies with, say, like ants or something, that's what would disprove evolution.
Right.
But I hate this idea that it's like, oh, well, common design, common designer. Okay, well, this common designer also managed to fit everything perfectly within a nested hierarchy. Where is my beetle with a mammalian ear? Right?
Exactly.
Well, where's my prokaryote with a eukaryote ribosome?
Yeah, or like a frog with an RNA-based genome, right? Like these are all possible. They're all very possible.
I mean, any organism that's not viruses with an RNA-based genome.
Yeah, but it's just okay. That's a bad argument. Anyways,
well, like they say it's a bad argument, but like, I mean, if you could have literally any other pattern, but you don't. So why is that? They have no answer for why there's a nested hierarchy in the first place.
Yeah, their best example against it is always convergent evolution. But even then, if you look at the synapomorphies of the convergence, they're never the same. Right? They're always like the duck-billed platypus. Oh, it has a duck's bill and a beaver tail, but it's actually extremely different. The way it's built, like the way the duck or the bill is built, is nothing like an actual duck's beak, right? And its venomous spike, the venom it contains isn't like any other venom found in nature. And its electroreceptors are more similar to echidnas than fish. So it's not this like mosaic species that God just sort of stitched together. It's just a basal mammal.
Exactly. Thank you.
Yeah.
What in the heck? Yeah,
That's why I was bringing that up in the after show the other day, like when he asked me about the eyes, and I was saying, okay, they're superficially similar, but they're not exactly the same. And he said, oh, but they couldn't have evolved more than once. I'm like, did you listen to what I
Why
said?
not? Wait, so was he talking about like mollusk eyes versus like
He was talking. He was talking about he was talking about all that and like eyes in general, and I was telling him that eyes have different underlying characteristics, like at the fundamental level.
Yeah.
And he just said, but they couldn't have evolved more than once. I'm like, they didn't
Why not?
evolve more than once. They evolved. They convergently evolved to look superficially similar, but they're not the same.
Yeah, I'm pretty sure most eyes are based on like sunspots and stuff that were in the last bilateralian ancestors, something similar to like a platyhelminth.
Right. Yeah, they all
really like arthropod eyes are nothing like the eyes that are found in vertebrates or mollusks. Vertebrates and mollusks actually convergently evolved to have fairly similar eyes, but the actual like the mechanisms behind it are completely flipped. Like so God, I forget what it is, but there's a blood vessel that connects into the back of our eyes, but the way it connects is weird because just the way our eye developed was inside out compared to the mollusk eye. Yep. So they actually have better eyes than us because they don't have a blind spot. Because
I don't think it's a blood vessel. I think it's the optic nerve, right?
Oh, yeah, that's it. You're right.
Yeah, but they all share some similarities because that's just common ancestry. But like at the basic level, for any particular convergent structure, it's going to be different in many ways.
Mm-hmm. Oh wow! Comments. The chat just exploded.
Yeah, well, just Athena and
like 15.
I don't think it was working before or something. I'm not sure. So, standing for truth. Yes, we are talking about the evolution part. Standing makes all this fun. I appreciate it. It's good to have a little fun in battle here and there. Yes, it is. We love you too, as you will see shortly.
Yeah.
The bear dog. Okay. Well, a bear and a dog both have limbs. They both have basically the type of cranium. They got a protruding mouthpiece. You know, so basically, when you see these different characteristics of different dogs, if you look at a bull mastiff compared to a chihuahua, you're going to see an extreme amount of variation. So if you want to draw a line,
that's because they were selected for.
Yeah, well, not even just that. Like all of the examples he's used so far have actually been closely related species. So his examples were human and chimp similarities, human and cat similarities, and or no, not human and cat, dog and cat similarities, and then dog and bear. Dog and bear are both caniforms. Dogs and cats are both carnivorans, and then humans and chimps are both hominina. I forget which specific sub family it is, but like.
Hello? Can you guys hear me?
Yeah, we can.
Okay. Yep. Okay. Yeah. Sorry. It's
tonight will be
for Jesus. Then. So hey, how you guys doing?
Good.
I don't even know how to fucking articulate how I am right now, but it's fine, I guess.
Oh, you're good. So, Tangelo, yes, we are going to be talking about biology here shortly, I think, but, a little bit. Smokey Smokey must have a stream right now.
Yeah, he's doing one of those open mic, fight nights or whatever.
Fight night. Oh, I like fight night. So, let's see, what do we got here? Standings. Okay, hello. Oh, Tangelo, my brother. Okay, nothing new. I can let this go. Okay, moving on.
And say that, okay, because a human.
Oh, C. Brown, are you there, brother?
Ears, got lips. They both got a little bit of. I'm sorry. Okay, so basically, it doesn't matter. It's not about drawing a line. It's about what mechanisms are there that can evolve a chimp into a human. You can't look at similarities because even if you look at DNA, you just go.
That's what does it, right? I mean, what is it?
What the DNA?
Well, yeah. I mean, what evolves a chimp into a human? Well, chimps don't evolve into humans, first of all. There's a common ancestor. But what evolves a wolf into a chihuahua? What evolved whatever the common ancestor was into a chimp and a human? It's the same. I mean, they understand that genetic changes can cause morphological changes, phenotypic.
By the way, I'm not a mod in your chat.
You what?
It's fine. It's fine.
You want to be?
Yes, I want a timeout standing. I'm joking. I'm joking standing. I love you. I love you.
Oh, we should time them out once in a while, just
How does
just
that
to
work?
keep up the.
Is it like
the
a
fun.
hockey rink thing where you just time someone out, like you put them in a little box for 30 minutes or 30 seconds or so?
Yeah, no, it's like 300 seconds, which totals 5 minutes.
Huh. I didn't know that was a feature. I've only streamed once, and I had like a total of 6 comments. So.
Can you put them on timeout for longer?
No, I don't think so.
I like the comments.
Can you roast them in hell for like 5 minutes? Wouldn't that be a cool feature?
I mean, that's God, isn't it? Only God can do that.
I'd like to be able to do that.
Wolf to Chihuahua is a reduction in allelic variation, though. Would Evo's claim chimp-human ancestor was the same?
Nope. And no, it's not. So wolf to Chihuahua and like wolf to Lassie, or what is it? What breed am I thinking of? What breed was Lassie?
I don't know.
Sheepdog?
Golden Retriever?
A border collie, maybe?
No, not golden retriever.
Oh, Lassie was a collie. Okay. So they found a gene, or basically, I believe there was a gene. I have to dig up that paper. There was an actual mutation that caused these longer noses in dogs, and it was not epigenetic. I'm sorry, standing, but your epigenetic modification doesn't give you anything at all.
He's also referring to a reduction in allelic diversity, which is fair.
Yeah. Okay.
But I would also say I would counter that by saying the populations. First of all, when you're breeding dogs, you're breeding specific lineages, right? So you're putting them through pretty much a bottleneck, right? You have 15 in a line that you're trying to breed straight down, and also, I mean, everyone's going to admit this. Genes are a lot easier to break than they are to build, and so when you're evolving something super super fast, you don't have time for beneficial mutations to accumulate and fixate, right? It's much easier for the deleterious mutations to accumulate and fixate. So I would say dogs probably have more like deleterious mutations that have achieved fixation, and that's resulted in their phenotypic differences compared to like wolves and stuff. But I don't think you can necessarily extrapolate that difference to all of life.
Right. Yeah.
Because when you're dealing with all of life, you're dealing with much larger timescales, and you're also dealing with much larger populations.
Right.
So, yeah.
And we'd have to go and look at the genomes of a bunch of different species of dogs. I still want to find it for mustard, for broccoli and cabbage and all that. I'd like to look at the genomes and start to straighten out how all that happened exactly. So, anyway, let's see. Okay, if I leave this on play, a raccoon just tore the fender off my car. I have to go feed them. Do you guys want to?
Well, I was gonna say, Tangelo said that he wanted to, or if we wanted to, he could come in and talk about macroevolution. I thought that'd be interesting.
Okay, let's have him in.
Yeah.
Yeah. Although I'm gonna run downstairs real quick and get a drink.
All right.
Okay, I'm going to send Tangelo a link. How the hell am I going to do that? I gotta. Oh my God! Somebody's at the door. Maybe it's a raccoon. I don't know. I'm worried. This is going to take me a minute, and then we're going to have to go feed raccoons. Let's see. Audio. No, there it is. Okay, I gotta get this address because it's in a different email. No, it was DoorDash. It wasn't a raccoon. That's good. We're good. Oh, where are we?
Hey, I have a question for you guys.
Go ahead.
So, would by any chance you guys be up for like more astronomy related stuff later, like whether the stream ends or not, or what do you want to cut it off later on?
We could, yeah. Okay. I know little about it.
Okay. Yeah, whenever you want to talk about it, like whenever the stream ends. But
okay. Tangelo link sent. You guys, who's still here? Anybody? Mutable, you here? Walker?
Yeah, I'm still here. Is Midnight still here?
Phil, yeah, no, I think he left again. Anyway, fill the dead air while I feed my pets.
All right.
Well, remember, when Tangelo comes in, you have to let him in.
Oh. You back, Walker?
I think Walker's still doing whatever he's doing.
Okay. Hey, when Walker gets back, I'd like to chat more about that particular mutation or whatever it was that they found. Scale changes in reptiles were equivalent to feathers. Basically, was it something along those lines?
Yeah.
I think that was pretty interesting.
Email sent. So, anyway, oh my, we have a thunderstorm developing. I might lose my power, but I have. What would happen if I lost my power? My monitors would go up, but my Mac would. You know, my Mac crashed today from StreamYard, and
wow.
I've never seen a Mac crash before, and it just went.
You're roboting.
It made a horrible noise, and it crashed. I think it was God's revenge.
Oh dear.
I think I StreamYarded my Mac to death because I did a couple of videos in a row, and then I had another one scheduled, and it was. So I'm not sure. I don't see how Tangelo yet, but we could play this a little bit.
Only look at the letters that align. So you really don't have a case. I'm gonna yield my time.
Okay, thank you so much for those points, C. Brown. We'll take it to David Neff, and you have 2 minutes of uninterrupted time.
Excellent, sure. So I want to begin by saying that commonality, creationists themselves are having a very difficult time determining whether or not some of these species are human or whether or not they're fully ape or whatever. Anthropogenesis, for example, has a few articles saying no, they're human, and another saying no, they're not human. ICR again. So this is creationists themselves. They're having a very difficult time with commonality and seeing all of this. I also want to say that the idea of chimps becoming humans is a straw man. Chimps do not become human. We do share, however, a common. Will you please shut up for a minute?
Every time he will interrupt somebody.
So again, chimps do not become human. We do have it.
Yep, he can't help it. It's kind of an OCD thing. Our last common ancestor evolving the humans would have been through reductions in allelic variability. Why? Or what types of changes evolved the ape-like creatures into man and independently into humans?
So I was actually typing out a response to that. So I think without knowing the genome of whatever our ancestral species is, we can't really know, right? There's no way of knowing. But I will say, so natural selection is a selecting force. It's a deterministic force that reduces allelic diversity, whereas mutations increase allelic diversity. And you have these two opposing forces that work against each other. So when you have enough time for mutations to accumulate and natural selection to select out things that are less favorable, then you don't really have an inherent reduction, right? It's an equilibrium, as Dr. Dan always likes to describe it.
Yeah.
Yes.
So there were probably, if I had to guess, obviously, because once again, we don't have the genome, there's probably a combination of reductive evolution where certain genes were lost, broken, whatever, and other genes were de novo, right, due to either duplications or just non-coding regions becoming coding regions.
Yeah.
But once again, I'm not super up to date on all that literature. That's just my best guess.
Yeah.
There are reductions in allelic variability. I don't think that is necessary. Or what types of changes evolved the ape-like creature into man? Mutations. All sorts of mutations.
Yeah.
I don't know. I don't see any necessity for an allelic reduction in any of that. So let's see. Where are we here? What do we got? We're at 159. We should rocket ahead to, well, we'll go ahead and play it. Oh, Tangelo's here. Hi, Tangelo.
Hi, good night, everyone.
Oh, yeah,
you
good night.
for having me here at your channel. It's a pleasure.
Yes, nice to meet you, my guy. And I'm also excited to see your conversation with Speed about abiogenesis later.
Yes. So I'm seeing that you guys are talking about common ancestry.
Mm-hmm.
Yep.
Okay.
Are you with that, or are you? I'm not sure if you're a young Earth creationist, or
Yes. I believe in a young Earth.
Okay, you do.
Okay.
So you wouldn't believe in common ancestry with humans and apes?
No.
Okay. Okay. So what shall we do? You want to?
Should we just do a free discussion, or?
We can, yeah.
I guess so.
Whatever you would like to do on Tangelo, because you're more or less the guest of honor right now.
You are the guest of honor.
Well, I thought when you guys were talking about common ancestry, you would have talked about the root of life, or the last universal common ancestor, and if that cell existed or not. And
well, I mean, quite obviously, it had to exist if things came after it, right?
Well, as I said, I am a young Earth creationist, so I believe that God created the various species or kinds. There was, in my understanding, a huge amount of initial injection of information and the ability of adaptation, and then there was a speciation of the second degree, where the species or kinds they adapted to the environment, and from there we had wolves becoming dogs, and cats becoming lions, and so forth. Speciation in a limited range, but not speciation from a universal common ancestor to apes and trees and humans and everything, and fish and everything else. That's in my understanding, that is far-fetched. I don't believe that this is possible based on my understanding on a molecular level.
Okay.
Why wouldn't I need to ask you a question? Why wouldn't humans start to evolve into like 20 or 30 different species, like cats and wolves and dogs and all of that? Like humans with, like pointed elf ears or different kinds of humans. Why wouldn't that happen?
Well, we see it doesn't happen. We know that there is a certain limit of variation of humans. We have Africans, Asians, and so forth, with each one with their individual traits, but it is in a limited range.
But do you believe that? Do you believe that Africans and Europeans are as different as house cats and tigers?
No, I think that the variation is much more limited in humans than in other species.
Why would that be?
Well, that's what we observe. That's what we see.
I do have a quick question, though. What do you think, like, this limitation is caused by, and where do you place that limitation at?
Yeah, that was my smart question.
Yeah.
Well, I understand that what causes biological architecture and form is not just mutations. That is the traditional view based on Darwinian evolution and the modern synthesis and so forth. It's a gene-centric view. And in my understanding, they started from 2000 so on to incorporate new mechanisms like epigenetic mechanisms. But the last endorsement by Feduccia, and so in 2008 he came up with his expanded synthesis, and they started to incorporate epigenetic factors. But in my understanding, there are many, many more. And in my understanding, what defines organismal form and complexity is a multitude of epigenetic mechanisms. And the main issue is information in epigenetics. And I have a list of over 23 different epigenetic codes and also signaling networks.
Okay, by codes, do you mean mechanisms of epigenetics, or do you mean like something different?
I have a link on my library. If Michael wants, he can.
How you're using the term like code in this case, like in regards to epigenetics.
Instructional, complex, codified information.
Okay.
Okay. So do you think that information is inherent, or do you think that it's encoded
there is non. Yes, there is non-genetic inheritance of information. Yes.
Yeah, but that only works for a couple generations, and that's only methylation. That doesn't. The
and
other code.
Huh? And acetylation. That's
the other. Acetylation is also inherited.
Mm-hmm.
Okay, but that's only on. That's.
There is much more. There is much more.
Much more, but it affects the histones, and it affects DNA, basically cell differentiation. It isn't passed on from generation to generation. That particular epigenetic change is not passed on. And,
well,
and the other ones are only passed on that we know of for about 3 generations at the most.
And also, if most of these differences were caused by epigenetic factors, I feel like that's a testable prediction, right? And using that prediction, you could see, okay, these specific alleles on these specific loci are hypermethylated in cats, which makes them stay smaller, but they're acetylated in lions, and that makes them grow bigger. That range of allelic variation isn't what we see. A lot of the interspecies differences are mutational, right? They're differences in the specific alleles and not the epigenetic patterns of those alleles.
Right.
Michael, I can link what I am working on right now. I did send you the link. If you want, you can put it on your screen. And then I will show you a list. What has to be explained is when you have, let's suppose, a unicellular organism, and you go from there to a multicellular organism, there are several things that must be specified. I list them in that link, which I will provide to you right now.
Okay, so let's choose one.
Would you like to put it on the screen, Michael?
I'm working on it. Working on it. It takes me a while to get it right. How we doing?
Okay, can you go up to the beginning?
Sure. Hang on a second. Let me. Okay, up to the beginning. The beginning is like right here.
Scroll up. Yeah, scroll up to the beginning of. Yeah, scroll up. Scroll up.
I'm going as fast as I can.
Faster speed.
Okay, here.
No, go up. Go up to the beginning. Okay, here.
Okay.
Here. You have a list here of 15 things. If you want to go from a unicellular to a multicellular organism, let's say you want a specialized second cell, and then you go to 4 and 8 and so forth, then you have 15 different things that need to be specified.
Okay.
And you don't specify that just by genes. There are many other things that have to be specified. There is cell phenotype and differentiation, cell morphology and shape, cell size, number of cells for each organ if you have a mammal or so, its specific function of each cell, the position and place in the body. This is crucial. Limbs like legs, fins, eyes, etc. must all be placed at the right place. How is each cell interconnected with the other cells? What communication is required to communicate with other cells, and the setup of the communication channels? What specific sensory and stimuli functions are required, and does it have to acquire in regard to its environment and surroundings? What specific new regulatory functions it has to acquire? When will the development program of the organism express the genes to grow the new cells during development? Precisely how many new cell types must be produced for each tissue and organ? Specification of the cell-cell adhesion and which ones will be used in each cell to adhere to the neighbor cells. There are 4 different classes. Programming of the time period the cell keeps alive in the body, and when it is time to self-destruct through apoptosis and be replaced by newly produced cells of the same kind. Setup of its specific nutrition demand. So you have 15 different things that have to be specified. If you have just a variation in the genome, how do you get all this all at once?
Well, you don't need all of it all at once. It's very much a gradient.
Well, phenotype and variation is due to how much some cells express certain proteins over others,
Right.
which changes their behavior, and it also changes how cells interact with their environment.
Yeah.
And how do you regulate that? How do you regulate that? Yes.
Sure. So you regulate that based upon what portions of DNA are allowed to be transcribed.
Yes, and how is that done?
There's proteins that bind both in prokaryotic organisms and in eukaryotic organisms that repress transition, but also allow for transcription to be more highly expressed based upon the factors that are binding with it.
Yes, but what is the mechanism that defines which section of the genome is transcribed?
Well, I mean, there's several, as you well know. I mean, a lot of it can be due to methylation, but a lot of it can be due to what the geometry of the DNA is, how the proteins are binding with that DNA at that particular point in time.
And I think another thing. Oh,
go ahead.
Oh, I think another thing is some of what we've discovered is that sometimes what genes are expressed depends on the concentration gradients of the different transcription factors in different organs as the organism develops. So like if you have a high concentration, you have these sets of genes being transcribed. You have a low concentration, you have these sets of genes not being transcribed, etc. So I think
what mechanism orchestrates the transcription factors and which sections of the genome is transcribed and which one is not? What is responsible for that orchestration?
Handled via metabolic control, right? I mean, metabolic, like proteins that handle metabolism, don't respond to overly high concentration or saturated at a specific point once they start engaging.
Have you heard about the gene regulation network?
Yes.
Yeah.
Okay, so that's the one which orchestrates the transcription factors and which parts of the genome is transcribed and when.
Right, but it's not the only thing.
Yeah.
Not the only thing. I can link you another site where I list the mechanisms which are, in my understanding, required for making organismal form and architecture. You can eventually put it on the screen, and then you can see the various mechanisms.
Okay, but I'm asking, would you consider it sufficient for differentiation to occur under the only condition being what proteins the cell are expressing and what abundance of proteins being expressed by the cell?
Well, of course, each set of proteins will be used in different cell types. But
that's not true of eukaryotic cells. Not all eukaryotic cells express the entirety of their genome.
Yeah.
That's not what I said. That's not what I said.
Right. You were saying how they express their genome, right? Which implies that they're using
in the human genome, we have about 200 different cell types. They express about there are 20,000 genes, and they are spliced through the spliceosome. And in human cells, there are 300 different kinds, and how the genome can be spliced. In each gene, 300 different protein species. And we have in each cell about 6 million different protein species. And each human cell has about 2 billion proteins. So you have an idea about the task of the human cells. So you can scroll a little bit more down, please. And these are the 17, there are more in my understanding, but these are the 17 main factors that are responsible for organismal form and multicellular complexity.
Mm-hmm. Yes, but also humans have way more like cell specialization than what's needed to be considered multicellular. Like I know of like very primitive sponges have only 3 different cell types, right? Instead of the 200. And of course, since they're eukaryotes, they still have a spliceosome, so they're still expressing a lot of different proteins. But that's what I'm saying when I mean, or that's what I mean when I'm saying that it's a gradient from hyper specialized cells that you see in most living bilateralians to the very unspecialized cells that you see in like cnidarians and sponges, periphery.
So let me just give a resume. In my understanding, what replaces evolutionary gene-centric mechanisms, that is allele variation, mutations, natural selection, drift, and gene flow, is pre-programmed instructional complex information encoded in various languages and communication through signaling through various signaling networks.
Right, but those are still genetic in some cases.
Right. Most of those are still genetic and protein based at the end of the day.
Well,
I'm curious.
In the list of there are many different epigenetic factors. If you scroll down, I'm listing 23 or 24 different epigenetic codes, and they are all relevant. Here. This list is all epigenetic codes, which are epigenetic. They are not. It's not genetic information. Mm-hmm. And only to give you an idea, just the glycan code on the surface of membranes is far more complex than genetic information. And,
right. Okay.
And science is just at the beginning to understand the code, to decipher the code, and to understand how that interaction between cells occurs. But the information is. Let me just give you an example, please. Yep. You have 3 billion nucleotides in the human genome, okay? But you have 37 trillion cells. Mm-hmm. And each cell has to be specified in regards of the list of the 15 different things, cell size. And one important thing is, how does the organism place each cell? How does it know where each cell has to be placed in the organism? You have 37 trillion sets of data, of information of each cell, where it belongs in the cell. So there is a huge discrepancy between the 3 billion nucleotides, the information in the genome, and the information required to direct each cell to its final destination and place, and timing, how many times that the cell cycle, there is about 40 times that is replicated. And if it's 41 times, then you have cancer and everything else. You have to know apoptosis, when the cell has to die and when it has to be replaced during development. So there is a huge amount of information which is not genetic. And what directs where these cells are placed in the body is the central nervous system through bioelectric codes. It's completely different. It has nothing to do with genetic information.
Well, right. I mean, it's due to the exchanging of voltage gradients and also hormonal signaling. A lot of that is.
Hormones, the bioelectric code. What directs it are neurons in the central nervous system, and science is just at the beginning to grasp how this works. They have not yet decoded the neuronal spike rate code. They don't know how to make the interpretation of the spikes. They are like a radio signal in phase and so on. So this is all things that science has no clue yet how to make the interpretation of all this. So the reality is that the complexity in biology is far greater than we have ever imagined.
Mm-hmm. Well, sure. I mean, I don't think even people even past Mendel's, even past where Mendel was when he kind of introduced the first notion of genes, thought that it was an extremely simple process. And people before we discovered DNA understood that DNA is something that has to be stored chemically, and even then that it has to be expressed in some way. I mean, I don't think necessarily people thought that this was going to be something simple. I mean, from a molecular level, yes, it's very complicated.
Yes, but my point is that the main tenet upon which evolution is based, that aleatorial mutations are responsible for that kind of complexity, that is just a hogwash pseudoscientific nonsense, which is 160 years old, which was proposed by Darwin when he had no clue about what happens in molecular in the dimension of molecules. And we know today that information is the main tenet, and you have to ask, where does all this kind of information come from?
Well, part of what you mentioned earlier with development, mostly, in some parts of development, is regulated by Hox genes. Particularly, I mean, salamanders are fairly well studied, and the fact that certain land salamanders, when you prevent a Hox gene from activating at a certain point of time, they start growing structures that look very much like gills of their aquatic counterparts.
Yep.
And part of it is the genetics that are associated with this, but also, I mean, the other things associated with it as well, direct kind of what type of organism something is going to be.
The Hox code is just one of many other different things that are required. Each cell needs to have the right shape. So when you have a blood cell, a blood cell has a completely different architecture and shape than a neuron, for example. We have neurons that are over 1 meter long, and that needs to be shaped. And how does that shape happen? How is the architecture, the shape of the cell, the mechanism? What is the mechanism that defines the shape of each of the 200 cells in the human body? And I can tell you, it's not genetic information.
Well,
no,
from the protein.
I would argue that a lot of the architecture of cells, different cell types, would be based upon what I said earlier, the types of proteins that are expressed and where those proteins are found in a cell.
No, that's incorrect. I have just studied this today.
Okay,
and if you want, you can link again to the previous page. And there I list the mechanisms which are responsible for cell shape.
Yes, the mechanisms are responsible for cell differentiation and cell shape and architecture and stuff. But ultimately,
it's not just proteins, as you said.
Well, when did I say that was the only thing?
Well, isn't the expression of those proteins what controls it?
No.
Okay.
Well, it's sometimes how
Yes,
much
people-
protein is expressed.
Is
Do you
that the
have
right
still-
page? Is that the right page?
Yes, you can go a little bit. Scroll down, please. Okay, it starts here. Okay, go a little bit up. Again, you can go a little bit up, further up. Okay, morphogenesis of eukaryotic cells, structure, and shape. Okay, so the shape of biological structures ultimately must arise from physical forces. So you have the tubulins, and these two tubulins, they exercise forces on the spindle, and if you scroll down, you will see there are various mechanisms. Science hasn't a full picture because it's very complex, but it's definitely not simply proteins. And what is essential is signaling. Whenever you try to understand development in biology, you will always come back to information and signaling networks, and that is also the case here. And that is a very interesting thing here. You can see the table of cell cycle regulators. There are 16 regulators which are kind of checkpoints during the cell cycle, and these 16 regulators, they are irreducible. If you remove one of these, then the cell cycle does not go through, and the cell cycle is also interlinked and is a part of the mechanism that defines the cell shape.
Okay, so what I was looking up one of these proteins, the geminin, the last one that's on the list. That is a protein.
Okay.
I would be willing to bet that all of these 16 factors that are listed, and there's more than 16 of them, are proteins that are determining or helping to determine cell shape and cell size.
These are regulators during the mitosis.
Right, these are regulatory proteins.
Yes, they are. But what is very important to say here is that the cell cycle is regulated by these 16 regulators, and if one is missing, you don't have a full cycle, and the result is cancer and all kinds of diseases, and in the end, death. So, how do you explain? How do you explain that? That is an all or nothing business. If you don't have full regulators, you don't have a functional cell cycle and replication. And how do you explain that through evolution?
Right. So the explanation with that is mutations that harm essential processes for cells, for instance, such as their differentiation and responsible for their shape.
No, if you have just a mutation, let us suppose that you have a mutation which produces in a miraculous way an entire protein of these 16 proteins, okay? If you don't have the other proteins, then you don't have a complete cell cycle because all the checkpoints are required to have a functional mitosis process.
Okay, I actually have a legitimate question because I'm not familiar with any of these. I'm not a molecular biology person.
Yeah.
But are there any multicellular eukaryotes that don't have these proteins? Like, you're saying like
every cell needs to be able to self-replicate.
Yeah,
okay?
no, I agree. But does every single plant that exists have this exact same set of 16 proteins?
Yeah.
Or every single sponge that exists or fungus have these same 16 proteins that control it?
Even unicellular organisms, they need to have the orchestration, the precise orchestration for their self-replication, and they need all these checkpoints and this regulation.
What paper is this? Looks interesting.
Okay. This is something from Tangelo's blog site.
Yeah.
Oh, and Tangelo's in here. Great. I need another headache.
No, you're good. Do you have a citation from like where this list can be found? Because I'm actually curious in looking into them, like sometime after the stream. Because I just don't know. like
Yeah,
anything about
I
them?
can link it to you. I have it at my site. Just a moment.
So where did you get this
Okay.
list? The purple one? Are you gonna? Is that from some book, or is that one of the links that you have embedded? Are you linking something?
I link always to my personal virtual library, where
Okay.
I investigate and then I collect this information and make it public. Okay.
Yeah, we're wondering what the source of this one illustration is.
Oh yes, it's also at the end of each post. I put the links to all the external sources and the science papers.
Okay.
I just did send you another link where you can access about mitosis and cell division. If you want, you can put it on the screen.
Okay.
Right. I mean, like my previous question was going back to, sure, I mean, if you delete one of these proteins, the cell probably isn't going to function. However, if one of these proteins has increased or decreased activity, be that it's transcribed more often or if it's not transcribed as often, that would change the morphology and physiology of a cell, wouldn't it?
Yeah.
So, I mean, the point being expressed here is that these 16 factors can't be deleted because they're essential processes to all cells. But if they change how often they are expressed, then potentially you can generate diversity within cell types.
Also, this is completely unrelated, but can I compliment on Tangelo, like on how comprehensive this blog is? This is
it's
incredible.
amazing.
Yeah,
it is amazing. How long have you worked on this?
About 5-6 years or so.
Wow.
That's a tremendous amount of work.
Okay. Look, you can just stop here.
Okay.
You see here the list, and each protein is the consequence if you don't have it.
Okay.
The 16 cell cycle regulators are essential. If one is missing, the cell cycle is not completed. Therefore, the regulation of the cell cycle is irreducibly complex.
Okay. Are there not other proteins that perform a similar function to these proteins?
Not that I know of.
Okay, so I mean, hypothetically, if there are proteins that perform a similar function to this, then it's not entirely necessary for these proteins to be active.
Well, what you need is, let us suppose, okay, in my understanding, for what I have investigated, these 16 cell cycle regulators are essentially in eukaryotic cells, okay? Let us suppose that rather than 16 in prokaryotic cells, you have 12 or 10 or so. But that will also be a minimum. And there is a threshold. If you go below that minimum, you don't complete the cell cycle. And then you don't have self-replication and no life perpetuation. And you need to have a fully functional set of proteins for the self-replication of the first living cell. And how do you go to that? DNA replication is extremely complex. You need about 30 different proteins to have DNA replication. And you do have, you need that set when life kickstarted. And prior to the first event of self-replication, there was no evolution. So how do you produce all these proteins to kickstart life if there was no evolution? If you cannot have a stepwise gradative evolutionary development?
Well,
that is a major abiogenesis problem. I can link you to my study on DNA replication, and you can have a look. I list there all the proteins that are required in DNA replication, and if you study it deeper, it's amazingly complex. If you look just into one of the proteins, like helicase requires 1,000 amino acids, and it has a performance of 10,000 rotations per minute. That is a fantastic molecular machine, and it has to emerge prior when life began. So my question is to you, If you don't have evolution to produce a helicase, how did it come to be?
Right. I mean, if you're talking about replication in a more general sense, I mean, there are several papers I've read which show that if you take a relatively random pool of RNA oligomers, and you impose it under a cold environment with the pressure to replicate itself.
Okay, I did link to my page on DNA replication. If you can put it on the screen, then I will show you all the proteins that are required.
No, sure. I mean, I know that there's a lot of them. I mean, it's just the question boils down to, was the first type of cell that replicated have to be the same way that modern cells replicate their information in their DNA?
Yes.
Okay, why?
Well, because that's the way how it works.
Okay, but so you would agree that the way cells take care of this process currently is efficient, though? Yes?
They're what?
It's efficient. The process that they go through in order to replicate information is efficient.
Yes, of course. Can you scroll down a little bit?
Sure. Right, but so you can have less efficient proteins that perform similar functions to this.
How far? Hang on a second. I just want to make a comment. I'm being very quiet. We are going to have a lot of fun on Monday. Oh, Tangelo, you and I.
Oh, yes, yes.
But right now I'm just your scroll bitch, your document bitch. So that's.
No, you can also give your input.
You know, Steve, if you
with
want
me.
to jump in at any point, feel free.
Oh no, I'm just enjoying this. We have enough of you guys to take over for me while I concentrate on what I'm doing here on the side. Okay, how far you want me to go down?
Yes, yes, you can scroll down, please.
Down further. Initiation. DNA helicase. Let's see. How further?
Yeah, that's the whole description of the replication process.
Okay.
You can scroll down and then maybe there is a list. Let me check. You can keep scrolling down. Okay, let's read the green part. Scroll up a little bit, please.
Okay. Can you see it?
No. Can you scroll up a little bit more, please?
Oh, more. Oh, that part.
The first one. Yeah.
The other green part.
One.
Okay.
Okay, let's read that question. Had not all three parts, there are three. Yeah, you have to read everything in order to understand what I'm talking about here. So, maybe we can just read at the initial of the page where I make the argument, so you understand maybe a little bit superficially what it is about. Can you scroll up again, please?
Yep.
Just to the top of the page, and I will read the argument. Just to the top.
Okay. So you make the argument of the original replicator.
Okay, the argument of the original replicator. In prokaryotic cells, DNA replication involves more than 30 specialized proteins to perform tasks necessary for binding and accurately copying the genetic molecule. Each of these proteins is essential and required for the proper replicating process. Not a single one of these proteins can be missing, otherwise the whole process breaks down and is unable to perform its task correctly. DNA repair mechanisms must also be in place, fully functional and working properly, otherwise the mutation rate will be too high and the cell dies. Just to give a parenthesis here, the repair mechanisms, they must go to precision of having just a mutation of 10 billion nucleotides. There can be one error, otherwise the error rate is too high. So error check and repair mechanisms had to be there when life began. Let's continue. The individual parts.
I disagree with that.
The individual parts and proteins require by themselves complex assembly proteins to be built. That means each of these proteins, they need to be assembled often they have cofactors as reactive part of the enzyme, and all this must be also assembled together with assembly proteins and chaperones and everything else. So these are literal machines which have also to be assembled like robots in a production line. Then the individual parts, assembly proteins, and proteins individually would have no function by their own. They only have function interconnected in the working whole. So if you have a helicase by itself, which unwinds the DNA strand, what is that's like a piston in an engine in a motor. If you have just a piston, it doesn't work at all. It only has a function if correctly inserted in the engine, and the same is here. A helicase has just function if it is inserted correctly and works in a joint venture with all the proteins to make the DNA replication process. Then the individual parts must be readily available on the construction side of the RNA replication complex, being correctly interlocked, interlinked, and have the right interface compatibility to be able to interact correctly together. All this requires information and meta-information, information that directs the expression of the genomic information for construction of the individual proteins and correct timing of expression, and as well the information of the correct assembly sequence. Evolution is not a capable driving force to make the DNA replicating complex, because evolution depends on cell replication through the very own mechanism we try to explain. It takes proteins to make DNA, replication to happen. But it takes DNA replication, the process, to make proteins. That's a catch-22 situation. DNA replication requires coded, complex, specified information and meta-information, and DNA replication process is irreducibly complex. Therefore, DNA replication is best explained through design.
Okay.
So the whole point here is that you need about 30 proteins, and the origin of those, you cannot explain it through evolution, because you need DNA replication in order to have evolution. So the only alternative is random unguided events, bringing up all this kind of complexity on a prebiotic earth to have a first self-replicating cell.
Did anyone follow that? I got a fucking headache now.
I followed it. I just don't really have a response because, like I said, my molecular biology is pretty weak. If you want to talk about phylogenies and stuff, especially of like animals, that's more my wheelhouse.
The truth is, if you don't have an explanation for the foundation of biology, and you don't have a naturalistic explanation for biology.
Tangelo, I believe in God now. You can go.
I agree with the point that he's making. But I just think, like, just because I don't have an explanation doesn't mean nobody has an explanation. I'm not sure if.
Well, let me give you an example. Okay.
Yeah, of course. Go ahead.
Let's take. I will screen another page. Then you can understand. Follow also by reading what I will say. Okay.
Mm-hmm.
Oh no. Video unavailable. Huh.
Okay, you can copy paste this link on your screen, please.
You want me to put this?
Yes, if you
in
can
the
do
chat,
that, please. Yes, please.
Okay.
I'm working on it. Yeah, somebody could put all those in the chat for me. That would be handy.
That's a lot of not genes.
Yeah, a lot of genes to put in the chat.
There's a lot of genes.
Hang on a second here. I got a little housekeeping to do here. Raccoons are breaking in for some reason. I have no idea why. What the hell I fed them. They're really irritating me now.
So is the Reason in Science website just essentially your website?
Yes.
Oh, okay. Well then, all of those links, I'm assuming, can be found just from like the home.
Okay,
Yeah.
Abiogenesis.
So
Come on.
I have a question for you on Tangelo. Just to clarify on something. If let's say in the hypothetical chance that evolution and modern biology cannot explain these mechanisms and how they came about. So I want to see like what other explanation would there be? What explanation do you think would be likely?
The alternative explanation, in my understanding, the correct explanation for multicellular organisms and body architecture and form is complex instructional codified information and epigenetic information and signaling networks on a holistic organismal level. That is my alternative explanation.
Right, but like you're saying that this cannot have originated through random events. So I'm asking, what other alternative is there for how it originated, basically?
An intelligent designer.
Okay.
So
how did the intelligent designer do it exactly, though?
I don't know. I give you an example. Let's suppose you think I want to move my arm, and then you move your arm. We do not have an explanation how that interface of your thought and the action of moving your arm works. We don't understand that interface. So I cannot explain to you how it is possible. Well, you can say, well, it's spikes in neurons, but I am a dualist, so I would not agree that it is simply a physical event. I believe that the mind and the brain and these are separate entities, but this is another issue. So basically, to answer your question, I don't know.
Let's back drive back down to the heart of like what you just said earlier, which is that you think that an intelligent agent would be necessary, correct?
Yes, I would like to give you a nice example. Can you scroll down a little bit, and we will just move on with Helicase. You will understand why. You will understand now why that is the. Yeah, just stop. Go up again, please.
Wait, Andrew, did you see the chat?
Okay, here, Helicase. No. Scroll up a little bit. You can leave it here. Okay, leave it here, please. Leave it here, please. Okay.
Andrew, did you see the chat? Emo said something to you in the chat.
Oh, we did?
Emo.
Yep. Emo's here, by the way.
Oh, what did he say? I don't have the chat pulled up. Sorry.
But he basically just said leave a comment on his channel. I'm assuming so he can like find your channel or something.
Oh, okay. Sorry, Emo. I forgot to do that. Oh, that was my mistake. Sorry about that. I have a lot of things constantly going through my mind.
Fair enough, my guy.
You there, Immutable?
Yeah, I mean, my point with Tangelo was, okay, so even if I accept the fact that, so you need an intelligent designer to do these things.
I will give you an example here with Helicase. We were talking about Helicase. Helicase is essential in DNA replication. It unwinds the DNA strands in order that they can be read and transcribed in RNA polymerase.
Yeah,
okay.
I get that.
So Helicase is a protein which had to emerge prior when life kickstarted.
Right, sure, I can accept that.
So Helicase is a protein which has 1,000 amino acids. Okay, Helicase are a class of enzymes vital to all living organisms. Their main function is to unpackage an organism's genes, and they are essential for DNA replication and evolution to be able to occur. They require 1,000 left-handed amino acids in the right specified sequence. Each of the 1,000 amino acids must be the right amongst 20 to choose from. So, as you know, life uses 20 different amino acids. In each of the 1,000 amino acids in each position, you have to select amongst 20 different amino acids. okay? And they can only be left-handed amino acids. Since on prebiotic Earth, there was no transaminase reaction to produce only left-handed amino acids. So they came in racemic form. They came left-handed and right-handed. But there is another problem. Based on scientific papers, they claim that on the prebiotic Earth, there were probably about 500 different amino acids extant. So you would have to make the selection. You would have to have a selection process amongst these 500 proteins and select the 20 that are used and functional in life. And you would have also to select amongst the left and the right-handed amino acids and just sort out the right-handed ones and keep the left-handed ones. That's the first problem. There is no selection process to choose the right ones. Second, you need to concentrate them at one assembly site. Third, you need a huge quantity of amino acids. Fourth, you have, if it is on the surface of the Earth, there was supposedly no oxygen, so you have anaerobic atmosphere, and then you have UV radiation which will destroy your amino acids. If the amino acids are on hydrothermal vents, you have another problem because you have no way to concentrate them at the same building site. So how did they emerge by natural processes? The chance to get them by random chemical reactions is 1 to the 20th to the 1,000. Actually, it's 1 to the 40 because you have to select also the chirality. So there are 10 to the 80 atoms in the universe, but the number of possibilities to have just one helicase is much, much greater than the number of atoms in the universe. Then you have just helicase, which is just one of the 30 proteins that you need to have DNA replication. So this is a killer in regards of naturalistic explanations of the origin of life. Done. There is no way that you get a helicase on a prebiotic Earth by unguided means.
How do you know that?
So I would question a few things. Number one, I don't think all 1,000 amino acids need to be exactly what they are currently for the protein to function. What needs to be conserved is whatever amino acids comprise the active site of that enzyme.
So put the number here. Put the number. What do you think, just hypothetically, what would be, in your view, maybe a minimal helicase?
You could probably do it with 50 to 100.
Okay. Then let me give you an example for a ribozyme, which is claimed to be the smallest ribozyme in existence. Okay?
God, I had PTSD from ribozymes.
You just scroll down a little bit and you will find it. Can you scroll down the page a little bit?
Yep. Let's see. How far? Oh, let's see. Do you have something on minimum?
No, you can go further down. I will tell you when to stop. Can you scroll down, please? There will come a sort of a table. Okay. Here, yeah. Okay. The simplest RNA proteins are ribonuclease, and it has 124 amino acids, and 19 of the 20 different ones are considered essential for life. Okay. So here we have the calculation to have the smallest ribonuclease, and the odds are at the last here 124. Is 2 to the 231th power. That is also far beyond the number of atoms in the whole universe. So even the smallest ribonuclease is too improbable to emerge by random unguided events.
Okay, we're kind of you're kind of repeating the irreducible complexity argument here, and as far as what the minimal helicase or method of duplicating DNA is, it's late. I guess we would have to start thinking about that. But I mean, we could go down any one of these trails and talk about the irreducible complexity of the eukaryotic cell. But you are looking at a eukaryotic cell, which is 4.2 billion years down the road. And it's kind of the same argument. So, I mean, we could go on and do this all night, but I don't think we would get any further.
I think we should leave some stuff in case you and Speed still have some stuff to go about.
Right. There's one point to consider here. I think you said like the minimal one, which is 124, needs about 19 or 20. And so that leaves you with about 100 that can vary.
No. What it says is that the minimal size, the minimal length is 124 amino acids, and that ribonuclease uses 19 of the 20 different amino acids.
Okay. So in that case then, assuming however many amino acids comprise its active site, which probably is no greater than 8 to 12, that means you have, 116, 112 amino acids left that can more or less vary. Because the majority of amino acids are non-polar, 12 of them are, and the other 8 of them are polar. And so I don't see why, in this case, the probability is considering the fact that each amino acid has to be exactly what it is. Although in reality...
Okay. Let's suppose that you can change these 110 amino acids and another configuration would confer a functionality, okay?
Right.
What function has a ribonuclease by itself laying around on early Earth?
Well, let's say, for instance, you generate a population of, say, 1 million molecules, and probably 80% of those will have activity and will have some catalytic efficiency to do something to that nascent change. So it might...
Well, the thing is that you need a very specific set of a proteome, and it is a minimal number of proteins in order to have all the functionality required to kickstart life. And you can take, for example, a cell, that cell, which has all the ingredients, all the proteins, everything, and then I tell you, try to reanimate it, and it will not be possible.
Well, right. So, I mean, part of the barrier in life, I don't think, is the metabolism. I mean, proteins are known to have ubiquitous activity, which means that you can throw random chains of amino acids together, put them in a solution, introduce some kind of native ligand, and they will have some type of chemical activity towards that ligand.
There are 3 main ingredients which are absolutely required to have life. One is the basic building blocks, the 4.
Right.
Then you need energy, and then you need information.
Well, sure. So I think
you need the 3 together.
Well, sure. So I mean, I think probably the most complicated part of that is finding the metabolism issue, because you can have all sorts of ways of getting oxygen into the atmosphere. You mentioned earlier that oxygen was very low on the early Earth. That's
okay. Let's consider then the problem of getting carbohydrates on the early Earth. Okay.
Right.
As you probably, I don't know if you know that, but there are 6 different carbon fixation cycles in life.
Okay.
And the most simple cycle is the Wood-Ljungdahl cycle, which requires 9 proteins.
Right.
Okay. And these proteins, they have a very high affinity to the intermediate product which they catalyze and which they hand over to the next protein. So the metabolism first hypothesis is that rather than enzymes, there were catalytic functions on clay and on metals and so forth, and that this somehow produced usable carbon on prebiotic earth. Now, Shapiro has clearly said that this is too unspecific, and the metabolism first hypothesis is bunk. It doesn't work. But let's suppose even that this would be not true, and that somehow by mechanisms that we do not understand, it would be possible without enzymes to fix carbon dioxide and get usable carbon on the early Earth. Now, you have a huge gap which is unbreachable to go from catalysts, natural catalysts producing carbon, to the simplest Wood-Ljungdahl pathway, which uses 9 proteins. These 9 proteins, they are extremely complex. If you take just the first one, it is one that has a molybdenum cofactor. And just to synthesize that cofactor, that is a molecular assembly line of extreme complexity. And these cofactors, they must be just right in their atomic structure. That is a kind of complexity we don't even understand how all that functions. Now, how do you have that transition from one to the other? That is a huge unbreachable gap, because you know probably very well how complex it is to synthesize proteins, the whole trajectory from the genome to the ribosome and to make these proteins. So that is a gap that you cannot breach. And in energy, you need energy to make proteins, but you need proteins to make ATP. So what came first? That is another chicken and egg problem.
So part of that is...
Wait a minute now. We've gone from helicases to metabolism.
We started on macroevolution.
Okay, so we're skimming from one subject to another, and you're kind of like adding things up. And I have a name for that, but let's not go there. On helicases, for instance, minimal helicase, the work they've done on abiogenesis on minimal helicase, I pull up 68,200 papers in Google Scholar. So should we read those tonight, or should we call it a night here? And
as I said, you can take the smallest ribonuclease, and it has 124 amino acids, but the problem is much deeper. The problem is you cannot even there were no, in the Miller-Urey experiment, they made only 12 of the amino acids which are used in life.
Okay, now we've ran across the football field 4 billion years the other way. You're talking about helicases, and you're talking about a highly evolved eukaryotic cell that's had 4 billion years. Now we're going back down to the amino acids. We can't be running back and forth like that. So we gotta pick our battles here. We gotta pick one spot, okay? And we're all tired, and you've put a lot of information on us, so we have to either pick something to drill into, or call it a night and digest this. So, but you've given us a lot to think about, and I'm really impressed with your blog, and I have been. I finally caught up to you as far as downloading papers. Just now. But to tell you the truth, it's past 9 o'clock, and I don't even know who you people are.
Oh, God.
You want to introduce yourselves one by one and kind of remind Speed who you are? Anyway, after 9 o'clock, Tangelo, my mind is just a little bit shut down. So, but anyway, very good presentation. Just really a lot to look at and a lot to think about. So I want to thank you. But I think we're overwhelmed for the night. So
Thank you for coming in and sharing your knowledge with us, though. It's been fun.
Yeah, it has, and really appreciative. And I and your blog is just wonderful. I discovered it, I think, a couple days ago, and I was just like, "Holy shit! This is a resource that is very valuable." So pretty cool. So anyway,
Yeah, I'm sorry that I jumped from one
yeah, we have to
issue to another.
Right. You had a lot to say, so it's fair.
Yeah,
no, it's good. I mean, we all get excited, and there's a lot to say here. So, I mean,
yeah.
But it's glad that we were all able to take something away from this, though.
Yeah.
If you've not understood something, you can ask me. There's no problem to explain what I'm saying.
Yeah, and I think we'll try to approach this from a more of a top-down level as far as the embryogenesis stuff that you started with on Monday, and we'll get into it and try to draw some pictures and explain what our differences are on this. But I have quite a few thoughts on that. So
yeah, okay,
okay,
no problem. You can select a topic which you have most interest in. I have also information about human evolution and so on. We can look on that too. I see that is a hot topic in various debates about common ancestry of apes and humans, so forth. So I have some material on that too, but I am more interested really on a molecular level because that is where the action actually happens.
Yeah, I am too.
The mechanisms that define organismal form are on a molecular level and not in paleontology and so forth, you know.
Yeah, that's primarily what I'm interested in. My friends here will tell you that I know nothing about humans.
Yeah, I actually am a biochemist, so I'm quite familiar with some of the molecular level of it. But with biochemistry, there's so much to learn about it. I mean,
and Walker is the guy for paleontology and humans, right? I think.
Humans, not really.
You don't like them either?
Walker, come on.
Okay, I like the. Oh, sorry. I do. So I would say I know a fair amount about human evolution stuff, but like Erica is probably the
Erica,
yeah.
How are you?
So
yeah,
a lover stat with Erica now. Here we go.
Yeah.
But yeah, paleontology in general, things like phylogeny, those are sort of what I'm interested in.
Cool.
Yeah.
Okay, I'm pretty burnt out.
Let's see if Standing shows up.
Well, I just downloaded about 300 papers, I think. Some of your YouTube links, Tangelo, some of your YouTube links don't work.
YouTube links to what? To my channel?
Sure. Yeah, on the blog, there's like 3 or 4 YouTube links that came up. The videos have been removed. Probably universities do that occasionally. But
yeah, of course, if I did put a YouTube video 3 or 4 years ago and then they remove it, I cannot check that all the time. So
yeah, I ran into a few. I didn't record which ones, but they looked like they'd be interesting to watch. But anyway, cool.
Should we end the stream soon, or?
Yeah, I think we could. I don't know. What about you guys? What do you want to do?
Are we going to Standing is coming in and see what that goes, or what?
Oh, do you really
Was there any more video you wanted to watch, or?
Oh, I mean, I think Standing will be also entertaining to talk to you.
Did he say he was stopping in?
He said
he said he was gonna try.
That was like 40 minutes ago, wasn't it? Let's see, 9:19. He said 9:19. It would be. Let's see, next.
He said it'd be an hour.
An hour, so that would be 10:19. So, up to you guys. Anyway, Tangelo, thank you.
Thanks, Tangelo.
Yep.
Okay,
Yep,
thank you guys for having me at your channel, and Monday we can continue our chat.
Right.
Of course. And they say we don't have productive discussions.
Yeah.
Yes.
I know.
And that we're not friendly. Okay.
All right.
Okay. Yeah. Bye, guys. Have a good night.
You too.
So now I'm interested in a helicase.
That's the only thing you're interested in?
Yeah. Yep.
There was
There's a lot of things that I want to look into. He was saying that the things that were irreducibly complex aren't found in anything else with cell differentiation. And I'm really curious about that because plants are really distantly related to us.
Oh yeah, yeah. And there's
I would have a hard time believing that all of them are found, but at the same time, of course, I haven't looked it up. So
I would have to say some of them are found because keep in mind, like when plants divide, they don't like their cell walls are shared essentially.
Yeah.
Like they don't separate cell walls.
No.
And that must be a hell of a process to get through. I mean, because
I was reading about that a few months back, but I can't remember too much about it right now.
there's an iBiology lecture on that thing, I think. Like plant cell wall composition.
Oh yeah, maybe that's where I saw it. Maybe I saw it on iBiology.
Were there any more videos or anything that you had in mind?
Well, I had this. The rest of, let's see, which one was it? The after show, I think it was. No.
Are you looking forward to your conversation with Tangelo yet, Speed?
Yeah, I am. It's going to be interesting. There are a
He
lot
seems
of.
like a nice guy. I think he'll say well.
What's that?
Oh, I said he seems like a nice guy. It'll go over pretty well.
Yeah, he is. Let me see here. Epic discussion. I'm trying to figure out where the heck I was.
I'm just wondering how many topics to bring up in that conversation.
There they are. There they all are. Okay. Yeah, there was some other stuff on this video that we could that we have not taken a look at.
Yep, you're still sharing Tangelo's blog though.
I am not anymore. I'm quick like a bunny.
He's learning. Oh my god! Take a photo.
Quick like a bunny, I am. I had to go battle raccoons. The mother brought her two little kids, and they were trying to. The fence goes down by the window, and they were trying to find a way in. Those little bastards
into the house,
into my backyard. I've got a cat fence in my backyard that's like 100% raccoon proof for almost a month and a half now, since they found a way to break in. But they were working on something tonight.
You ever think you're installing like an electric fence?
What's that?
With electric wire.
Oh, that's a good idea. I was thinking about dynamite.
Yeah, I was gonna say I feel like it's gonna evolve into like a barbed wire fence with like lasers and shit.
I feel like if you give these things enough time, they're probably going to develop Tony Stark level technology. Just have like Iron Man suits fly over.
Well, it's cool. I'm proving evolution with my raccoons.
Yeah, like I said, I'm pretty sure I've mentioned this before, but there's some really interesting like studies on the urban development of raccoons. So
yeah,
because they're getting better at like problem solving. Yeah,
we're talking about that in our private chat, I think.
Mm-hmm.
So
hey guys,
anyway, let's see. Is this the right video? It is. Okay, so where are we? We're on 159. So there were a few points here. 159, 204, 2. Yeah, I guess we can just go on and listen to a little bit of this. Tell me if the sound. I think I clicked the sound. Or no.
I think Andrew was wanting to say something real quick though.
Oh, Andrew, what's up?
Oh, I was wondering if you guys still want me to wash some astronomy down your throats.
Yeah. Sure.
Oh my God, we could do that if you want to.
Sure. At this point, I was hoping we could just do a free discussion, if that's all right. I'm kind of burnt out.
Sure.
Fair enough. We
Yeah,
do
you want
to? I mean, if you want to as well, because.
Do you want to end the broadcast and do it?
Sure. Whatever you guys want to do.
Yeah, we could do that.
Going to give Standing an opportunity to come in and embarrass himself. I mean, have a good conversation.
I'm afraid that would go long and turn out badly.
Yeah, I feel like I'd have more to say against Standing though.
What's that?
I said I feel like I have more to say to Standing than like Tangelo though.
Yeah, probably. I just want to talk to him about genes just once. Come on.
Oh, I do not want to get into the whole created heterozygosity thing until we watch the rest of this video. There are some bookmarks, but I don't really want to do it tonight.
Okay.
Oh, come on.
Fair enough.
Yeah. if we're going to do astronomy, I think that we should do that offline and go ahead and call it a night. And I've got bookmarks here, and I will try to do a better job. I want to work some more on this, but I want to show you why Standing for Truth's heterozygosity, created heterozygosity, is such bullshit.
Yeah.
But anyway, that's going to take a little bit more brain power than I have tonight. I do not want to get into it and get into it badly. I want to get into it thoroughly. So,
yeah.
And
did Tangelo just wipe you out?
Downloading all those goddamn papers did. I bookmarked. Oh, what did I bookmark? Something like 200 tabs, and I was afraid that Chrome was going to crash. 200 or 300 tabs, I think, with just the papers that he has backing all that stuff up.
Mm-hmm.
And
was that like from all his sources, or is that just
all of his sources that he listed? I basically got them all. I've got them all bookmarked. Now I've got to figure out how to get the papers and which papers I can get. A few of them were PDFs, but not many of them. So it's going to be a challenge. There'll be some work. Right. Anyway, so we're going to end the broadcast tonight. That was cool. So anyway, I'm ending, and then we will talk offline.
All right.